Welcome. Welcome to the GRC America's Regional Scientific Webinar on COVID-19. I'm going to ask my colleague Roberta to start the presentation. So this is a series of seminars that the GRC America's region has launched on COVID-19. My name is Alejandro Adem. I am president of the Natural Sciences and Engineering Research Council of Canada and also a professor of mathematics at the University of British Columbia. Now let me give you an overview of the aims and activities. Roberta? First of all, what is the GRC? The GRC stands for the Global Research Council. It was established in 2012. It is a virtual organization comprised of the heads of science and engineering funding agencies from around the world. The collective vision of the organization is that research and innovation play a leading role in providing the solutions that society needs to overcome the global and interconnected challenges of our time. Our mission is to work in partnership to create the conditions in which international research cooperation and collaboration can thrive. What are the objectives of the GRC? Well, first of all, we want to act as a forum for funding agencies to share data and best practices and discuss issues of common concern and mutual strategic priority. Secondly, we want to actively facilitate and promote bilateral and multilateral collaboration between our participant organizations to support the global science enterprise and address global grand challenges. We want to champion a more equitable, diverse, and inclusive future to harness the global diversity of talent. And fourth, we want to maximize the value and trust placed in science by society and to engage and communicate with society and the public. Now, the regions we have are Americas, Asia Pacific, Middle East / North Africa, Europe, and Sub-Saharan Africa. This event is for the Americas region. And in particular, we're talking here about the event highlighted in yellow on research infrastructure related to COVID-19. The countries of experts are Argentina, Brazil, Canada, Chile, Paraguay, Peru, and the United States of America. We've had a series also on health systems and education coming up. And what are the objectives of these workshops? Well, they are to provide an opportunity for scientists to discuss their research with international colleagues in a workshop format. And what are the expected outcomes? Well, we want to foster international research collaborations and strengthen interagency networks within the Americas. Now, today we have some excellent experts. You see here their affiliations and their photographs, an outstanding group of people. So what we will have today is a short presentation from each panelist, and then we'll have questions and a discussion. So the panelists will be speaking in the following order. So first of all, Wilbert van Panhuis from the NIH, who is director of the Office of Data Science and Emerging Technologies of the National Institute of Allergy and Infectious Diseases, followed by Rafael Tapia Limonchi, who is director of the Instituto de Enfermedades Tropicales, National University Toribio Rodriguez de Mendoza, followed by Diego Stalder, who is a professor, artificial intelligence at Asunción National University, then followed by Claudio Gutierrez, professor in the computer science department at the Universidad de Chile, followed then by Pablo Labrado, researcher and director of the Center for Urban and Regional Studies, Konicet in Argentina, followed then by Helder Nakaya, senior researcher, Hospital Israelita Albert Einstein as well as adjunct professor at the Emory University School of Medicine in the US. And finally, my Canadian colleague, Professor Prabhat Ja from the University of Toronto, Canada Research Chair at the Dalla Lana School of Public Health, and founding director of the Center for Global Health Research at St. Michael's Hospital. And I guess we're ready to start then with a presentation by Professor Van Panhuis. After the presentations, we will have an opportunity for questions and discussion. Thank you. Over to you, Professor Panhuis.
Thank you. Thank you very much for giving me the opportunity. I'll share my slides here. Okay, can you see the slide?
Yes.
Okay, good. Thank you, and thanks for the organizers for giving me the opportunity to present here, and it's a pleasure. I will talk about our vision and ideas at NIAID about data discovery and integration for infectious and immune-mediated diseases in the context of the research infrastructure topic of today. Just an introduction. So I am Wilbert van Panhuis. I'm the director of the Office of Data Science and Emerging Technologies at the National Institute of Allergy and Infectious Diseases at the NIH in the US. NIAID, as you may know, is one of the 27 institutes and centers at the US National Institutes of Health. The Institute mission is to conduct and support basic and applied research to better understand, treat, and ultimately prevent infectious, immunologic, and allergic diseases, and also to respond to emerging public health threats, including pandemic threats, as you may know. The Institute has awarded over 6,000 research grants and contracts to the extramural, to the research community in last fiscal year, 2021, at a total value of about $5 billion US. In addition to the funding of research in the community, the NIAID has also an extensive number of intramural research labs of research that is conducted inside the Institute, as well as the Vaccine Research Center. Current strategic priorities of the Institute, of course, are to invest in advancing research, basic research and translational research on SARS-CoV-2 and COVID-19. Also, to work towards a universal influenza vaccine, work on medical countermeasures for emerging and re-emerging diseases, infectious diseases, and immune-mediated diseases, work on HIV vaccines, prevention and treatment measures, and look at the prevention and treatment of immune-mediated diseases, especially immune tolerance issues. So that's about NIAID. I'd like to acknowledge the team members of my office, as well as colleagues. And I have to say, I was just starting at NIAID in the last, about 5 months ago now. Previously, I worked at the University of Pittsburgh in the US, and also had collaborations with the GoFair organization in Europe. And so a lot of the work I'm showing here is based on work from all of these colleagues together. The infrastructure, research infrastructure vision that we're laying out is really that is the perspective with which I give this talk here today. It's the infrastructure, the policies, and also the culture that is involved in improving, finding, accessing, and using data for biomedical research. That's what our office is looking at. And the vision is that researchers and programs can easily find the data they need for their research, that it's clear who can access a data set and how, that it's also clear when, where, and how data are supposed to be shared or expected to be shared by the research community. Also, data can easily be used together with other data to answer new questions, and data are preserved and used again and again as new scientific hypotheses emerge. Even if the PI has left a lab or program officer left the program, there is the continuity. So that's the vision of where we want to get to. And as we all know, data is a pillar of the scientific method, not just now, but for a long time already. This is a map of John Snow that many people may know from 1855 in London. During the cholera outbreak, he was showing all these deaths. Each bar is a death due to cholera at an address in London here at the Broad Street area. And he showed that a lot of these deaths occurred around this pump. And then that made him believe that the pump and the contaminated water was the source of the cholera outbreak. And so data have been used for a long time for science. And I'll show you some examples now for more recently how we use data to get impact and look at scientific results. This is a project I did at the University of Pittsburgh with our colleagues there, Project TICO, to improve data access and use through data curation and standardization. There's a lot of data that's difficult to use. It takes work to standardize and integrate the data, but then it can be easy to use for researchers. Here is what we did with all of these data from books and tables online that were not easily analyzable. After we prepared the data, we could look at these patterns here. Here is you seeing measles in the US. This is weekly data from 1928 to about 2010, showing incidence rates of measles going across the country here at the country level, annual measles outbreaks. Then for each state in color coding, within red, high levels of outbreaks, high incidence rates, and in blue, low incidence rates, and in black, missing data. And you can see the different states here listed, 50 states. Pennsylvania is here. That's where Pittsburgh is, where I'm currently even located, even though I'm still working at NIH. Here is the outbreaks. You can see a lot of outbreaks. But then here in 1963, the measles vaccine was licensed. And you can see the drop of measles after. It's a very strong difference in before and after the vaccine. And we calculated that 35 million cases of measles were prevented since 1963. And so the data, just showing the data, had a very dramatic impact in showing the history of this vaccination program in the US. And you can make similar images for all over the world because the impact has been the same. And we did the same for many other diseases here: smallpox, polio, diphtheria, pertussis, rubella, mumps, hepatitis A, and published this paper showing 100 million cases have been prevented since 1924. And then the impact occurred when we released the data to the public so that anybody could analyze the data through this website. You can see here the number of users going up from 500 to over 5,000 currently. And a lot of papers that came out since 2013. Here you can see all the different research papers, not by us. These were papers published by other researchers using the data that we made available. And so it has accelerated science to make that data available in this way. We had the same experience during the SARS-CoV-2. As we all know, the genome sequence of the virus was shared very rapidly. In January 5, the WHO report was published about the outbreak in 2020. In January 10, the sequence was published on the website here, and also later in GenBank, and the paper came out in February 3. So that sequence here in that I kind of minimalized here has led to an enormous amount of advances, as we know. The structure of the spike protein was identified, and then the vaccines, Moderna and Pfizer vaccines, and others later, and many other countermeasures came from that sequence that was published. So that's a really strong example of how data sharing has accelerated these countermeasures. But the challenge is that the data landscape is complex and siloed. A lot of different repositories exist for data, for different pathogens or different types of data. We have repositories for tuberculosis, for HIV, for malaria, but also for multi-pathogens, for clinical records, for genomic data. And then there is data on journal supplemental data, on journal websites, university websites, lab or personal websites, or some data not even discoverable online at all. You can call that dark data because nobody knows that data exists. And so the siloed repositories that don't talk to each other and the data without a home makes the landscape complicated, and it's hard to use data from different places together. And for researchers, that can be a bottleneck. And so during COVID, the same kind of landscape very rapidly emerged. There was an unprecedented amount of data sharing, but there's also an unprecedented amount of data systems developed very quickly that don't always talk to each other and are not always connected. There were case report data on various different places. There were clinical data in a lot of different repositories and systems, and then serology, intervention, behavior data as well. And I would say that the limited interoperability between the data sets and the limited metadata and the lack of a centralized data search portal can slow down research and discovery. It's hard for a researcher to know where to go for the data. And data cleaning as well is a bottleneck to scientific progress. Lots of surveys have been conducted now showing that it takes a lot of time and effort to clean and prepare the data before you can do the analysis. And we would want to get to a place where we can minimize that time, that intelligent and smart researchers with enormous analytical skills don't have to spend 80% of their time cleaning data, but they can spend the time on the analysis part. There are also non-technical barriers. We did a review in 2014 showing 20 barriers to data sharing in 6 categories. There are the technical issues I talked about, but there are also motivational issues around scientific credit, potential criticism, economical costs and damage potentially from sharing data or information from countries, political issues, legal and ethical as well. So there is a whole constellation of considerations to make when thinking about advancing data sharing and data use in science. And so then we have the FAIR principles that many of you have already seen probably. And I think they are a very useful set of principles that lay out a blueprint for research infrastructure for what we can do. They're not too prescriptive, but they leave a lot of interpretation and they leave a lot of, but they're kind of foundational building blocks to look at the findability of data through unique identifiers, metadata, etc., accessibility, interoperability, and reusability. And if we create research infrastructure that is following the principles, then almost as a result, the data will become more connected and more interoperable and easier to use. The FAIR principles are also geared towards really teaching machines how to use data, especially artificial intelligence and machine learning, but also for humans. So my colleague Barrett from the Netherlands says, does the machine know what I mean? That is what the metadata and what these principles are for, metadata for machines. But to make metadata for machines, we need to first make metadata for humans so that humans can understand them as well, human researchers. And so metadata is key to FAIR research infrastructure. Just a little depiction here that there is a data file and there is a metadata file. The metadata are describing the data about what is the title, who are the creators, how can you access this data, what is the geographical scope, etc. And that's described in the metadata about that data file. And then the metadata can be stored separately from the data, but point to the data and help discover this data. And the metadata can be copied and distributed and indexed and searched. But the metadata can also point to the terminology used in the data file so that it becomes interoperable. And so the idea is there that metadata gets indexed and searched. Here are data sets in the data landscape. Each data set has metadata, becomes discoverable through search. All the data sets without good metadata are not so easy discoverable. And then what we want to do is get the good metadata for all of these data sets, become all discoverable, and then the researcher doesn't need to do little work to try to put it all together. That's the ideal vision. We're not there yet, of course, but that's a vision where we want to go to. And so NIAID, our institute, is building a data ecosystem that does some of this to enable the search and discovery across repositories, to connect repositories to a centralized portal for searching here and for analyzing the data. And also bring then the data without a home, data in all these other places that are not in a repository, into the repositories, and then develop policy guidance for data access and sharing. And then the idea, and we already just recently in September this year awarded the contract here to start building this system. And so that's an exciting development to hopefully make some improvements in the landscape. And so lastly, what I would say, since this is a workshop about collaboration, I hope we can all collaborate on this research infrastructure. We can try to move as much research data and rich metadata into the established repositories. NIH has a policy developed for data management and sharing for our grantees and for the researchers at NIH to work towards that. Use the existing repositories where possible, no need to reinvent the wheel. And then develop and sustain FAIR compliant data repositories. So there are funding opportunities that have been published to do that through the biomedical data repository knowledge bases. And then we can all collaborate to include relevant data repositories into the ecosystem. The ecosystem will be open, and so any data repository can be put into the ecosystem and then be discovered with all the other data sets together. And then I would say promote the secondary use of existing data, and there is funding opportunities for that as well. And then participate in our data sharing science webinar series and other data science funding opportunities. And these are all links in the slide. If they will be shared later on, then people can just go to those links and explore for themselves what may apply to their situations. And with that, thank you very much. Happy to take questions if there's time, and otherwise, I'm looking forward to the discussion.
Thank you very much, Wilbert. I think we're going to leave the questions until the question session at the end after all the talks. So that's okay. I believe that's,
yes.
Yes. And we'll move on to our next speaker, who will be Professor Rafael Tapia Limonchi. Professor Limonchi, please.
Well, thank you very much for the introduction. Good afternoon, everyone. Thank you to Konicet and the GRC organizing committee for the invitation. I would like to talk about, well, a very young institute, the Institute of Tropical Disease here in Amazonas, Peru, in the northeast of Peru, and what we are doing to contribute for the R&D development here in Peru. Well, the Institute is a scientific institution oriented to enhance the research, development, and innovation here in this northeast side of Peru. It's 1,000 kilometers from Lima. It's a very ancient community, region. We are trying to create new knowledge about the tropical diseases using high-tech tools of molecular biology and biomedicine. So, but why Amazonas? Well, the major of the research here in Peru for tropical diseases is always oriented to Iquitos, that is close to Amazonas. But Amazonas, for many time, for many years, had a problem for neglected from the health system, and with a high percentage of poverty. So, but by the location of Amazonas, that is a subtropical location, there are many endemic tropical diseases that are circulating, like malaria, dengue, leishmaniasis, among others. Further, the population, more than 50% of the population lives in a rural area with low basic resources, in absence of drinking water, tap water, sewage. We have native communities in a province, Condorcanqui. It's around a population of 35,000 people with high levels of poverty. So the history of the institute starts in 2016 with the approval by the Ministry of Education of Peru for a public investment project. Then in 2019, by a funding of Konicet in a project for reincorporation of investigators, or for Peruvian investigators, we start activities of research. For the next year, we'll start the build-up of the new building and the installation of all the equipment for the correct function of the institute. And well, we invite all the researchers, all the investigators that want to interested in tropical diseases to give a view of this institute. We have 4 research lines. The first one, genomics and molecular epidemiology, that seeks to look for how is the behavior of the transmission of many diseases during outbreaks or during surveillance, trying to look for gene markers for viability or for drug resistance, for example, also. The other line is biosensors and POC devices to develop efficient POC devices. And the last one, the cell cultures, trying to take advantage of the mesenchymal stem cells to face some injuries that produce infection diseases, for example, inflammation. About our technology, we have all the stuff for molecular biology and next generation sequencing. We are part right now of the genomic surveillance for SARS-CoV-2 national program. In the next months, will arrive a peptide synthesizer for enhance the POC and RDT's device development, and for explore new candidates for vaccines or new biologics. The other one is a Luminix 200 that will allow to implement multiple diagnostic platforms for outbreaks, for a rapid answer in outbreaks that are very frequently here in this area. For us, the networking is fundamental. Since the beginning, since the start, all the investigators promote the institute, try to get some strong collaborations with some universities from USA, Europe, and in South America also, and with some institutions like Cayetano Heredia University and National Institute of Health here in Peru. Well, we have a very short life. We have 7 published articles in Q1 and Q2 categories. We got 4 projects from Konicet funding for more than $1.2 million. And in the region, we support to the regional health authority, the directorate here in Amazonas, to implement a molecular diagnostic for SARS-CoV-2. Before that, there were not any molecular laboratory here in Amazonas. Until now, there are more than 18,000 samples that have been done, and always with the genomic surveillance for SARS-CoV-2, trying to see what variables of the virus are circulating here in Amazonas. Our main concern about the SARS-CoV-2 is how is the behavior of the disease in a background with other infection diseases. Here in Amazonas circulate dengue, malaria, and on other viral diseases. So, for example, we in February 2020, we found a falciparum malaria transmission after 15 years that non-autochthonous transmission. So it was important, but then in March, after the lockdown, until now, all the surveillance and control programs are very dramatically affected by this. there's no in research, and our proposal is to implement control studies to determine the impact of the co-infection. No, that we are blind about what happened here in native communities or in the urban population. Finally, I would like to present the new building, the infrastructure that we are building here in Amazonas. It's a 4 plants building with 2 wings. The left wings is for the wet lab activities, and the other one is for the administrative issues. But the interesting part of this building is in the 3rd floor. We have considered 2 rooms independently. One is a biosafety 3-level laboratory for tackle these biosafety issues about the handling of high transmission pathogens. And the other one is a GMP room for the manufacturing of some biologics or new advanced cell therapy, for example. This model, it could be the first one model of this infrastructure here in this area in Peru. Thank you very much. Thank you for the organizers, and please do not hesitate to consider the Institute of Tropical Diseases a good partner for research. Please visit also our webpage, and thank you very much.
Thank you. Thank you very much, Rafael. I think I see some questions for you already in the chat that you might want to address in our question and answer session. Now we move over to Professor Diego Stalder, professor from Asunción National University. Diego.
Hey, can you hear me?
Yes.
You can see my presentation?
Yes, we do.
Okay, great. Thank you for the invitation. And today I'm going to talk about the capacity building in the metrology to support the development of medical devices in Paraguay. This is part of a project founded by Konicet from Paraguay and implemented with several institutions, engineering faculties, our space agency, and our metrology institute, national institute. Okay. The COVID-19 pandemic has placed enormous strain in our public health systems. And early in the pandemic, a shortage of ventilator was motivate engineers from different universities around the world to develop new prototypes of mechanical ventilators, and also car makers developed their prototypes. We saw several prototypes in Latin America, Brazil, Peru, Argentina, also in Europe. And here in Paraguay, we started a challenge in March 2020, and several groups of engineers, technicians, scientists, and medics developed their prototypes. Here I have some picture of the prototype developed in 1 week. Some of them are very simple. And this challenge motivated several companies to go further and develop ICT ventilators. And they invested to develop 2 prototypes for critical care applications. And then we had the Konicet call for projects in July 2020. And with this call, 38 projects were approved, about $1 million in total. And in this scenario, there were several prototypes. We have a few prototypes on the faculties and another prototypes from the companies. The companies, the velocity was since they have more resources, they were faster. So they did also trials with animals, and so in that scenario, we saw that there was a missing capability in the country. There were no laboratories to validate those prototypes, and this is why we wrote a project to build a medical testing lab to evaluate these national ventilators prototypes. But the specific objectives was to determine the efficacy and the safety of the prototypes, build a testing lab to validate them, and to assess the quality of their designs, and also to develop a low-cost test platform for other areas that they have not the infrastructure to validate them. And also we defined the test criteria. We hired some bioengineers to test the prototypes following international standards, and then to develop the bioengineering in the country. So the team was in the team we have several engineers with different areas of expertise. I am an electronic engineer, but my research area is applied computing. But we get the help of bioengineer and also with other medics. And from the space agency, we have a team another. And we have also collaboration from other countries, from Peru. Sandra, she has a laboratory, a metrology laboratory there, where Masi was tested. Michael from Vanderbilt University, and also a startup incubator from Argentina. We have also the help of metrology, National Metrology Institute, and this was the team. So we started improving all the space to perform the test. This was the improvements of the laboratory, and this is the layout now. We have the space for the efficacy test, for electrical safety test, and we build this cell to evaluate the pre-compatibility of these prototypes. This is under development because we build with the low cost resources, and because in Paraguay we don't have an echo chamber, and there are only in Argentina and Brazil. So to develop new technology, it will require more funds to do the electromagnetic compatibility test. So for the verification and validation, we consider international standards, and those standards requires the use of gas flow analyzers and variable test loop to mimic different pathologies and to evaluate the prototypes and the different critical conditions of the patient. And also we evaluate the electrical safety, measuring the resistance and insulation under different fault conditions. Here I have some example of the efficacy test. In some cases we have good results, and in other we have errors in the physiological values provided by the prototypes. So the developers improve their designs. This takes time because we have to do several combinations and several hours and several repetitions to have the accuracy also of the test. Another product of the project was a low-cost tester, because these gas flow analyzers are too expensive for our universities. It costs about $10,000. So we designed a low-cost tester using a Raspberry Pi and medical grade sensors and data acquisition system. And we designed a structure to integrate all the to integrate all the subsystems. And we validate this with a calibrated gas flow analyzer. Here we have one example of the interface where we can see the three main variables of the ventilation system: the pressure, the flow, and the volume. This is an open source ventilator. We are publishing this design. We are going to submit the design to a journal now. The electromagnetic compatibility is to evaluate if the noise of the environment of the medical equipment can affect the new device, and also to evaluate if the noise generated by this device can affect other devices. So the low-cost solution was to build this gigahertz transverse electromagnetic cells, which is we designed this and we performed some simulations to optimize the dimension in order to generate the uniform electromagnetic field according to the standards, and we build these cells. This is the cell that we built, and we have here the absorbent that we imported from, in this case, from China. So the future projects that the future activities that we are going to we are planning is to have more human resources to provide services to the hospitals in our country, to have the traceability of the medical equipment, and also considering also ventilatory support equipment and electromedical equipment with this device equipment that we bought with the project, to perform also electromagnetic compatibility tests. And we are planning also to incubate startups to develop new medical devices. Now we have a new fund to develop one ECG, portable ECG, a low cost. We have now a fund to create a startup to design this new device. And the idea is also to collaborate with other universities and laboratories to develop new medical devices and improve our testing protocols. So thank you. This is my email, and I will hear the questions.
Thank you. Thank you very much for your presentation. We'll move over now to Professor Claudio Gutierrez from the University of Chile. Professor.
Okay. Thank you very much. Let me share the slides. I hope you are hearing me.
Yeah, all good.
Yeah, it's okay. Can you hear me? Good. So I will talk briefly about the data challenge that our experience with the COVID-19 outbreak. I belong to Universidad de Chile and to the Millennium Institute of Foundation of Data. that is a research institute here in Chile that is devoted to this to research about basic issues on data. So the COVID pandemic has given us a lot of experience and lessons in order to work, to perform, I mean, to know how to deal with digital data. I will present, sorry, I will present more abstractly. I will go directly to the agenda. This was one of the questions in the agenda, how to strengthen data system, digital infrastructure with attention to the collection, storage, maintenance, governance, distribution, and use of data. If you can see from here, you can immediately observe that here there is a classical vision of data, the traditional vision of data. In fact, these are the pieces of this classical cycle of data. What is happening today is that we are dealing with a more complex scenario than this one. That is a more complex scenario in the sense that there are more organizations, more type of data, more volumes of data, and more interconnection of data available. So here in these questions, there are involved data, what's really, what we understand by data today, data system, digital infrastructure and collection. And I will just touch each of these things with an eye in the COVID data. So let me first remind you the source of healthcare big data today. So essentially the data comes from wearables today with apps and medical devices, from electronic and medical and health records of the population, insurance providers or state data, other clinical data, obtained genome and research registers, social media, and knowledge on the web. So this is, this is the source, this is the material that we have to deal with today. Let me also remind you the issue of scale. We can talk about human scale of data when we are in these four areas of the byte is a character, the kilo is a written text, the mega you can associate it with image, sound, giga with video. But today we jumped, and you can see that all these type of data are processable by humans. Yeah, in fact, long ago, before the digital world appeared in the 60s or the 50s, 60s, we dealt with all this type of data by books, by films, by music records, and so on. The problem today, or the issue today, is that we have data at the level of tera, peta, exa, and close to zeta. Terabyte, you can, associate it roughly with the size of the Congress Library in the USA, not considering videos and images. Peta is a big data center. Any big data center has petabytes at least. Exabyte, all spoken words, and today there are about 150 exabytes of healthcare data only in the United States. And so you can consider, when we talk about health data, we are in this range. When we talk about COVID data, we are in this range here. So far beyond human scale, so far beyond the techniques, the classical techniques of dealing with data. Here you have, this is an image from 2015, but the predictions are roughly correct. Yeah, and just to let you know that here, look, this piece is sensor and devices, social media, voice IP, and enterprise data. You can consider that health data involves about half or a third of the data in this range. And look here, we have volume in exabytes. Yeah, today, 2020, we have about 9,000 exabytes of data, a little bit more. I will jump this. This is a slide by Hans Moravec, and it's very interesting to see that, I mean, the capacity, just to stress this idea of beyond human, the capacity of the human here is about 1 billion megabytes. The capacity of human and processing instruction per second is about 1 billion. And here are our animal friends that you can recognize. And any application today, any simple application is far to the right here and above here. So it's far beyond the human capabilities of processing it. So we have to rely in some sense in other devices. So this is a picture, a rough explanation of this. This is the world before 1950, okay, in science with a very strong impact here, and you can think of science as a body of knowledge that captures in some sense all the algorithms and all the processing things that were done by the humans through instruments. Yeah. But today we have something like this, yeah, where data and software has taken a little bit an autonomous drive here. Yeah. The humans, in some sense, control. Fortunately, we still control here, and we are going to control this. This is no doubt. But the science as such, I mean, the body of knowledge is playing a role, but directly on the data. I mean, the main, let's say, the main actor today is becoming data instead of the knowledge of the sciences. This is the classical data life cycle, as I told you. See, classical collection, relevance, classification, handling, storage, transmission, manipulation, release, backup, retention, and publication. Yeah, and I just showing this slide because it's interesting to see here they put the risk level: high, moderate, danger in semaphore semantics, and you can see where are the red: handling and storage, manipulation, and alteration. And strangely enough, there is no other red point, but I would put a red point here in release because of privacy concerns. Yeah, that is have become a real hot topic today. The novelty today regarding this, I mean, this cycle is really the scale of the data, at the techniques of collection, usually implicit. I mean, the explicit recording of data when somebody asks you about data explicitly. I mean, you know when you are giving your data is a matter of the past, okay? So almost all the data that you are giving from your identity, bioidentity or psychological identity, is usually captured implicitly via social networks, via devices, via sensors that you are not aware of. So this is one of the novelties and the challenge. And then the processing and predicting of ML, I mean, all the techniques of artificial intelligence, but essentially neural networks in some particular architectures that are taking the lead in the processing and prediction of data, all fed by data. When we speak about data system in the questions in the agenda of the seminars spoke about data system, I would add ecosystem, and it's not about only data. Yeah, this is a diagram by Puneet Kishore, and you can see that really the ecosystems where the data lives, yeah, is being done by raw data, but it's being done by APIs, application interfaces, by repositories of different things, by literature, the classic literature in publishing, binary software, and biobanks. Yeah, this is very important. Look, this is material thing. This is a material thing here. Yeah, materials explicitly here, and there is analytics also. But the interplay of all these things is what one has to, to design when thinking of a system, a data system, and thinking in data infrastructure. That infrastructure is not anymore like in the decades in the 80s, 90s, hardware and software. It's much more than that, and is interrelated in some very tricky, I would say, and complex ways. The digital infrastructures involves classical.
I just ask you to speed up. I just ask you to speed things up a little bit because you're running out of time.
Okay, I will finish in the next one. So I hope that this is known to everybody. I will skip this one, and I just will put the title of the challenge, okay, that I think deserve to be considered when thinking about the infrastructure of data. Thanks.
Thank you very much, and we'll certainly be able to revisit that during the question session. Just have to keep on moving through with our speakers. So our next speaker is Pablo Labrado, Director of the Center on Urban and Regional Studies, Konicet, Argentina.
Hello. Yes, I will put my slide. Okay. You can see it. It's okay?
Yes.
Yes, thank you very much. Well, thank you very much for the invitation to this webinar. I'm going to refer briefly to the results of a research work we carried out with another researcher on the capacities of biomanufacturing in Argentina, one of the topics that was in this webinar as a question. And then ask ourselves about policy issues of complementarity at regional or international level. Our perspective is more global and a social science perspective. Is an economist perspective, developing country vision with the questions of a country like Argentina, that like other countries of Latin America have some minimal research infrastructure, but it has also problems of health sovereignty or the health autonomy, with trade deficit in vaccines, biopharmaceuticals. That's the issue we will afford in this presentation. So the two questions we try to answer are firstly how to build capacity in biomanufacturing and life science to ensure biomanufacturing facilities designed with resilience processes and equipment, and how to cooperate and share infrastructure investment internationally or regionally. We think some lessons from Argentina recent experience that perhaps help to answer this question or open new discussion. The first question, briefly, we can say that Argentina has health biotechnology research and development infrastructure that is with some development for a developing country. No, there are more than 80 9 Konicet institutes in areas related to health and biotechnology. And we have in Argentina more than 3,000 researchers in biological and health sciences. Recently, we have incorporated good laboratory practices that are one of the bottlenecks we have in research facilities to go down in value chain to production. In terms of clinical studies, there are more than 18 companies that they have their own clinical capabilities, and they are complemented by a dozen of private clinical study laboratories. So the research network have some development, but the problem is that when we see some economic indicators as pharmaceutical industry balance of trade, so export, less import, we have the problem of pharmaceutical increasing trade deficit. Argentina have expanded the social the access to pharmaceutics, but industrial capabilities have not go in line with this expansion of biopharmaceutical and pharmaceutical needs. So this poses serious problems of policy. Is a problem of health policy and a problem of industrial policy, industrial and technology policy. So, but we can say that when we analyze different kind of companies, we have in Argentina, well, before that, I can say that one of the explanation of the deficit, of the increasing explanation of the deficit, is the increasing of biopharmaceutical products that are increasing their share from only 55% to more than 35% of total deficit. So more complex and high price biopharmaceutic are explaining the increasing of deficit. So we have to say that this deficit is not evenly distributed between different kind of firms. We can say here that in Argentina there are 71 biotechnological firms which produce biopharmaceutics or import with some final stage of fill and finish or of packaging of biopharmaceutic, and more than the big part of this deficit is explained by multinational companies and national companies, so the national pharmaceutical affiliates, diversified national pharmaceutical firms, or dedicated biotechnological firms. They are all superavitary. No, there are no deficit at national firms. So when we see the experience of these firms, we see that this capability, the biopharmaceutical, biomanufacturing capability, is associated to some policies that were developed since 2010, policies of Technology and Science Ministry. We have supported biomanufacturing capabilities. So here there is an issue very important of industrial policy, the central role of national firms. Biomanufacturing capability in Argentina. This is a process, not long process, but we have some results. When we see some numbers of these capabilities, we have mapped with a survey to all these firms, and we could see that dedicated the national firms, there are this kind of firms, they have they have all biomanufacturing capabilities. This is measured in liters. No, so Argentina have a 13,000 liters capability. Is not little. This is remarkable because Argentina is perhaps one of the poles of the nodes of the region with possibility of a regional network with production capability. This can be illustrated by some experiences, recent experiences, where Argentina have, for example, make cooperation between Argentina with another an Argentinian firm, make some cooperation or with Brazil's firm to technology transfer of manufacturing of biosimilar. Then there are cases of technology transfer from Russia to Argentina to fill and finish. Also, this is the case of also the same thing that make the biosimilar technology transfer, but in that case, the transfer of vaccine AstraZeneca capability of manufacturing. So Argentina produces the active pharmaceutical ingredient of a big part of AstraZeneca bio vaccine, which is producing for all Latin America. So this give us some lessons. This process are not all good news because we have the problems of a developing country. The first problem we can say is that perhaps we can make a lot of effort in research, in development of first stages, but there are problems of regulatory complementarity. There are high costs of clinical development. In the case of vaccines, we could see that there have been problems to access of critical supply from global suppliers of filters. And now we have a situation when Argentina, Mexico in this case, have expanded a lot the biomanufacturing capability at national scale. So this overcapacity, when pandemics decrease the needs of this kind of perhaps these kinds of vaccines, if there is windows of opportunity to new vaccine developments for neglected diseases as Chagas and others, or for biosimilars. So there is a big avenue that is open for the region to cooperation, not also in research and development, but also in production, and particularly in product development. That is not the same as research. So the main conclusions is that despite the trade deficit in pharmaceutical products, Argentina is characterized by an incipient scaling and manufacturing capability of active pharmaceutical ingredients, which have allowed it to rapidly diversify toward the manufacture of recombinant viral vector vaccines against COVID-19. The experiences of productive complementation show absorption capacity both for the reception of technology transfer in the manufacturing and formulation phase of vaccines, and the capacity for technology transfer to other countries, because Argentina also have made technology transfer to Brazil. And the deepening of the experience of regional complementation poses several challenges that advance not only to research development, but also in local productive development, which implies advancing in funding pools of clinical phase 3, number 3 clinical phase. A complementation in regulatory requirements is very important, and it's very important also the division of labor in the different phases of the productive chain. So in research and development, in manufacturing, and in fill and finish. So well, thanks for your time. This is my email. I finished my presentation. Thanks.
Thank you very much, Pablo. Thank you. Now over to Helder Nakaya, Senior Researcher at the Hospital Israelita Albert Einstein in Sao Paulo. Over to you.
Thank you so much, Alejandro. I'm so happy to be here. Let me share my screen. Okay, so today I'm going to talk about network science from Zika to COVID. And what do I mean by network science? Well, we've all witnessed that during the COVID-19 pandemics, someone in China, for example, can generate a nice results, and then they can put the data in a server in UK, for example, and someone in Canada can write a nice review with a question. And here in Brazil, we may use this question and to be inspired to perform some analysis, and then we can get public available data and perform the analysis. And if all goes well, the results can be applied somewhere in India, for example. So that's what I mean by network science, which is the people collaborating through exchanging data and also exchanging expertise. And of course, for that, you end up using the infrastructure of everyone. And in Brazil, we do have a lot of people working on Kovids. These are some of the, of course, researchers that collaborate in different papers. And most importantly, they did not start collaborating during the COVID-19 pandemics. They were already collaborating even in the Zika epidemics. And of course, between the Zika and COVID-19, we had other epidemics in Brazil, such as chikungunya, yellow fever, measles, and we always have dengue, malaria in few places, right? So all this can only be done, could only be done with the support of funding agency, including FAPESP, that supported many researchers here from Sao Paulo directly or indirectly, even if they are outside Sao Paulo. So in our lab, we also receive a lot of funding from these agencies to study different kinds of infectious disease and vaccines. So my work actually started when I was at Emory University in US, but we kept working in different diseases such as influenza, chikungunya, pneumonia, schistosomiasis. And we use this approach that's called systems biology and network medicine, which utilize a lot of public available data, so we don't need to generate new data for that. And what do I mean by network medicine? So let's take two conditions, a healthy person and a sick person. And we can use high throughput technologies and state-of-the-art equipments to measure the biological feature within a condition. And then we can do a lot of experiments to show how these biological conditions, biological components are connected. So this is the network construction part. And then when you compare the networks from different conditions, so that's a network medicine. And that's what we have been doing for a disease such as Zika. So in this paper, a lot of collaborators help us to understand what's going on in the brain of babies that die with Zika and microcephaly at genomics level, transcriptomic levels, and proteomics level. We also work with several researchers in Brazil to investigate the molecular mechanism of chikungunya and why they cause arthritis in some of these people. And of course, when COVID-19 hits us, we start working completely on COVID-19. So these are the 30 papers that we have been working, collaborating or leading articles in different areas. Actually, we use mobile applications, we are developing computational tools, we work with diagnostics, epidemiology, and of course, the systems biology, which is our expertise. So for computational tools, I don't have time to show this because the goal here is to show what is the research infrastructure needed. And what we try to do here is to create open source applications and computational tools that were useful for epidemics or infectious disease. And I select these 4 papers here to talk a little bit more because I want to show how can you do a lot of good science without having to generate new data. So for example, in this first paper here, we are actually wondering why people with different comorbidities will have higher chance of developing severe COVID-19. So that's the question we ask. What are the molecular factors associated with severe COVID-19? So this is just to show a time frame. The World Health Organization declared a COVID-19 pandemic in March 11, and we submit the paper to publication on March 27. So in less than 1 month, we were able to collect public available data ask the questions and then do the analysis that is now have more than 200 citations. So that's just to show how powerful it is to invest in nice collaborations and use the existing research facilities and data. So what we did was to get in the public repository data from transcriptomic of the lung tissues of patients that have comorbidities associated with severe COVID-19. And then we found the molecular factors that were common to all of them. And then one of the findings that one of the genes that were there was the gene that encodes the main entry receptor for the SARS-CoV-2 virus. So the hypothesis that the paper suggested is that people with different comorbidities have higher levels of ACE2 in the lungs. So when the virus goes into the lung, it's easily find another cell to infect. So that makes easier to virus replicates. And that increased the risk of developing severe COVID-19. So that was what we published here and showing also the mechanism behind, trying to identify the mechanism behind. So in this paper here, we collaborate with people from Unicamp. And what we did was to reanalyze public available data of single cell transcriptomics. So each dot here is a single cell. And then we look at monocytes and found that evidence that they were infected by the SARS-CoV-2 in the lungs. And then Pedro Vieira's team at Unicamp showed that you can infect monocytes with the virus, but if we increase the dose of glucose, the virus also increases the replication. That's then we look back at the molecular level and we see glucolysis, and then people from Unicamp tested this by blocking different parts of the glucolysis, showing that, for example, only in this example, that if you have a vehicle, the virus replicates. But if you block here, the virus does not replicate anymore. So what they show is that the virus needs glucose and the HIF-1 transcription factors to do the infection. And that's maybe why diabetes is an important comorbidity associated with severe COVID-19. So this paper here, I chose to talk because that was derived from this repository of FAPESP. So FAPESP collects laboratory test results from different hospitals and diagnostic companies. So we analyzed over 4 million laboratory test results. This is all public. And then we could see, because we have so many people with COVID-19 and over 400 analytes, we could see what are the changes that occurs in different genders and in males and females, if they have or not COVID-19, and also across age. We could see that depends on the age and the gender, these factors change a lot. And of course, it changes if you go or not to the ICU. So this last paper here is just to show that we were investigating a completely different disease, which is Barrett's esophagus. And what happens that after we analyze this public available data, again, it's free, we come up with genes that were upregulated in most of these studies. And one of the genes is the gene that encodes the receptor that the virus needs to enter the cell. So that suggests that Barrett's esophagus could also be a comorbidity associated with severe COVID-19. And then when we start to look at the pathways, we see that the acid pH, the intracellular pH, was somehow driving this expression. And I'm gonna just show here the validation where we collaborate with people from Unicamp that validate the findings for us. So after we do the analysis with free data, we sent to these people to verify if this is true. So what they did was to culture monocytes with different pH and then show that the ACE2 expression increase. But then they also infect the cells, showing that when you have acid pH, the virus replicates more than when you have normal pH. But is there any clinical implication on this? So how can we find who are the people that have Barrett's esophagus or gastritis or reflux? Well, they usually take proton pump inhibitors, omeprazole, pantoprazole. So what we did was to collaborate with people that already analyzed cohorts of COVID-19 patients. So we separate people that were taking the drugs from people that were not taking the drugs. So that's the indirect evidence that these people here were treating diseases such as these ones. So the interesting thing is that once we analyze this cohort from the North region, we see that people that do not take the drugs survive more than people that take the drug. And when you adjust by diabetes and hypertension in old age, you see that the risk of dying of COVID-19 is increased if you take the drug. And then we replicate these findings in another cohort in the Southeast region with another set of collaborators that are also financed by FAPESP, that also confirm that people that take the drug have increased levels of chances of dying of COVID-19. So to conclude, I'd like to show that I gave you examples of how you can use, how you can perform low budget, high quality science by using data that is already generated. But the problem is that, of course, to do the experiment, someone spent a lot of money. To store the data, someone spent a lot of money. And of course, to train the researchers to do this analysis, a lot of money was also invested here, right? So this is an example of the hospital that I work now, which is they are finishing this building and they are going to invest on working on infectious disease, which was not traditionally a type of disease they were interested, but now they are going to support a lot. And of course, FAPESP, if you take all these researchers that are here now producing about COVID, actually they were derived from investment during the genome project, where FAPESP invests a lot of money in training people in the state-of-the-art omics technology at the time. So I hope FAPESP is now going to invest heavily on single cell analysis, because that's going to be, I think, the next phase. So what I meant by low budget is actually these parts here. And we can apply this to COVID-19, but also we can apply to any other disease that may come and will come. And with that, I'd like to acknowledge people from the lab. And of course, thank you all and sorry for speaking too fast.
Thank you. Thank you. You really packed a lot into that. Thank you very much. Moving now to Professor Prabhat Ja from the University of Toronto.
Right, let me just give me 1 second to share my screen. Oops. Sorry, just 1 second. Right, I hope you can see the screen. Okay, I'll take the silence as a yes.
All good.
Good. Okay. I believe I'm the last speaker, and I'm always careful about standing between people and their lunch. So let me go quickly. And I'll start by actually giving you my conclusions, which really are about a commentary on the science response, which might be needed for COVID. So first is to very much recognize that now COVID is a disease of the unvaccinated. So the top priority, and perhaps the only one, is to vaccinate all adults in the world. And I'll illustrate that quickly by showing you the consequences of unchecked pandemics in India and Latin America, and a little bit about the economic and social consequences. Going forward, can we prevent future viral waves? Well, we really don't know. We're in this era, we have to define and live with endemic COVID, and which vaccination will be the key to reducing hospitalizations and deaths. But I will argue that what we also need is to think about launching a global adult vaccination program, strengthening surveillance, and increasing both epidemiological and clinical trial science in some countries. So just as a comparison, how bad is the COVID pandemic versus the great 1918 pandemic flu that affected worldwide, perhaps causing 100 million deaths? Well, the short answer is that even at the very peak of the US or Latin American death rates, the COVID death rates were far below what was observed in the influenza peak. And much of this peak was because of, well, in the UK was because of soldiers returning home from World War II. But you see, there was already an earlier peak followed by another peak. And in fact, COVID or influenza persisted not just for these 2 years, but went on in smaller waves for many more, and eventually was transformed into a more seasonal flu patterns. So that's the good news, that our death rates are nowhere near what is feared as catastrophic. But the bad news is that in places where transmission has been uncontrolled for good stretches of times, there's been an extraordinary doubling of death rates during those peak pandemic months. And just to show you India data, well, the problem with trying to sort out data in India is that of the 10 million deaths that occur, most do not have a death certificate and most occur at home in the rural areas. So you need alternative sources of data, which we put together. And this paper currently under review at Science shows the following. In India, COVID was the main contributor to the substantial excess of deaths. And the actual totals were at least 3 million deaths, which is 7 to 8 times higher than the officially reported. And as context, WHO's worldwide estimate is 5 million deaths from COVID. So the Indian numbers alone would substantially increase that. And broadly, the COVID death rates in India are those in Latin American settings where there was uncontrolled transmission, such as in Peru. So these are some of the results from India. So what we've relied on is a national polling survey, and it's got a very simple comparison. In the dotted line, you see the monthly patterns in pre-pandemic baselines in 2019 and what happened in 2020. Well, COVID deaths were slightly higher, but non-COVID deaths were reported being slightly higher, but this is probably misclassified COVID deaths. And here you see in 2020, the situation, 2021, in the first few months, a huge increase in COVID deaths, but also in non-COVID deaths, again, mostly reflecting COVID or misclassified COVID. So overall, about 5% of houses reported a COVID death, and that would be consistent with roughly 3 million deaths. Another source of information is to look at medical facilities, which are all over the country. They're not representative, but they're national. And the same comparison, the dotted is the baseline of 2019 deaths. Here's the September, August first viral wave in 2022. And here's the catastrophic increase in May of 2021. The government has not released data for sensitivity reasons from June onwards. And this pattern is very similar to what has been observed in Peru, where, again, compared to the average deaths in the previous years, there was a huge spike in total deaths. And that was broadly paralleling the reported cases. So overall in Peru, which has done an outstanding job of providing good updated data on causes of death, death rates approximately doubled. So that's the really catastrophic news that we have to think about in the context. But deaths are certainly not the only metric. We have now good evidence of long-term morbidity that could follow mental health in youth and young adults in particular. Young adults and jobs have been particularly affected. The education statistics are staggering. It might mean that worldwide children have lost in affected areas half a year of schooling, which will affect their lifelong earnings. And in birth rates, there's even suggestion of an extraordinary decline in the pandemic. People don't stop dying during the pandemics, but they do stop having sex, and therefore birth rates did substantially fall. Now, the extraordinary good news is that almost all of the vaccines that are widely used are remarkably effective, and they're more effective against severe disease than they are, which is shown in the vertical axis, than they are against any infection, which is in the horizontal axis. So if you look at all studies, they basically are above that line, meaning they work better at preventing severe disease, that's hospitalizations or death. And it doesn't much matter about the variant. There's some how well it works by the variants does vary, but it's consistently better for deaths and disease than for infection, and simply also by vaccines, and also whether they reported early or late. So vaccines have to be the heart of what has to be done. So what does this mean in terms of thinking about the epidemic? How does this end? The answer is it doesn't. The the only way we can think about it is as many people on Earth as having antibodies and T cells that can ward off. And this is much better achieved through vaccination than recovery from infection. And we will move to endemic eventually, but we have to know what that means. So will it be that SARS becomes more seasonal like the flu, recurrent and seasonal? I believe so. But we don't have good enough evidence saying, well, if 2 or 3 doses of vaccines provide lifelong protection, it might be more like measles with seasonal, with not a strong seasonal component. A seasonal component, particularly a flu type pattern, is likely, and we know that it'll have disproportionate impact in mortality in particular age groups, the elderly, immunocompromised. So how do we move to endemic? Well, the ethical and scientific approach is to vaccinate the world as quickly as possible. The most important benefit is that it prevents the variants of concern to develop in variant factories from low vaccination rates. And that includes parts of the US, for example, with low vaccination rates are basically factories where variants of concern can occur. And something better than Delta, at least from the virus's perspective, might emerge from those uncontrolled or limited controlled settings. The world's progress on vaccines has been, as we all know, extraordinarily mixed. Portugal sets an enormous standard of more than 80% of its adults being vaccinated. Canada has done well. United States still lags behind. Brazil, after a slow start, has come up. But overall, the large populations, particularly in Africa, are still the ones that are lagging behind. And this is for a very simple reason that rich countries are competing for limited supply with the WHO COVAX facility. So the WHO COVAX facility, and this is their estimate, has suggested that in high-income countries, there's 155 doses per 100 people now available, mostly from bilateral agreements. So this is richer governments being able to secure deals. In low and middle income countries, only 58 doses per 100 people, and in the low income, only 9. And that very much has to rely on the COVAX part, I mean, less so on, has to rely particularly in low income settings. The International Monetary Fund has a plan that would cost $50 billion to vaccinate 60% of the world by 2020. And the key here is to focus on the green population, or the ones in arrows, which are about 1 billion people that are in other low-income countries outside of China and India. China and India effectively have scaled up their own vaccine manufacturing capacity, so they probably will meet these targets. But the rest of the world outside of high-income countries are left on their own. So that includes Latin American countries also. And what is needed? Well, the IMF has very much estimated a huge increase is required in, sorry, not sure what that is, my apologies. Much stronger science is needed for dealing with the COVID response. As we know, it's unvaccinated populations, but that does not remove the need for ongoing randomized evidence, including of what are the possible ways of introducing booster shots. We're relying too much on limited data from Israel, for example, and not enough on direct randomized evidence. Mortality and case data are needed, and certainly a global genotyping effort to track variants. So let me show you one example of a study in Canada where we've tried to follow the national population, which is a very simple national study, which involves sending roughly 10,000, 9,000 Canadians a kit at home, which they, it's basically a mailed kit that looks like this. We mail these to about 10,000 Canadians and ask them to take a stab of blood at home. And they do a dried blood spot, put 5 circles onto a paper, and then they mail it into our central labs. And we've now done 3 rounds of this. And what have we found? Well, cumulatively, the percentage of Canadians infected rose to 6.5% from about 2%. So Canada's had generally a modest epidemic. the prevalence rose nearly everywhere, but in Atlantic Canada, which had better control, the prevalence was lower. Visible minorities had a larger increase. And the good news is that among those that had definite infection, up to 7 months later, there's a durable immune response. So this is shown in this graph that using different antibody cutoffs, roughly over 7 months, you have stable response. Now, what we're doing with this study is to turn it from an infection cohort to a vaccination cohort. So the phase 1 studied roughly 9,000 right after the first viral peak in Canada. Phase 2 covered the peak of the second one. And phase 3 was just been completed, and that was launched when peak vaccinations were done in Canada, shown in red. So we'll be able to comment on vaccine and natural induced immunity going forward. So now let's put COVID mortality in the context of the big causes of death globally. And yes, COVID has been extraordinarily disruptive. At its peak, 50,000 deaths a week were occurring, but most of these were above age 70. And it is worth noting that roughly 20 million deaths in middle age, 30 to 69, occur. So from vascular deaths, there's about 100,000 a week just in middle age, and from cancer, 80,000 a week just in middle age. So we should be thinking about efforts that increase the epidemiological science to these routine conditions, not to the dramatic. And one of the important cross-cutting pillars is to much strengthen mortality science. So to do so, we've launched the Global COVID Mortality Database, GCAM, at the University of Toronto. And we're setting up a new platform called AI Code, which has that clever acronym that you can see, which would try to get individual death records widely available for researchers to use for COVID and other analyses. Let me come back to randomized trials. The WHO vaccine trial design is something I believe should be implemented in many countries. The reliance on administrative data is a strategic mistake because we might well be missing the opportunities to really learn what types of combinations of vaccines and what dosing schedules and others are very effective, particularly if we're going to have if we're going to think about a flu type pattern of periodic vaccines. That in turn demands a global adult vaccine program. And broadly, the goal should be to vaccinate all 5 billion adults, as we do with the extended program in immunizations for kids. And that could both do the COVID vaccines plus routine ones, flu, pneumococcal vaccines, HPV, and newer vaccines as they come along. This would require expanded supply and economic incentives. John Bell has written about this recently in The Economist, as we have in The Lancet. Pair that with a global surveillance facility, which really needs to think about having robust systems to monitor data. So let me conclude. I think COVID now is very much not just a past challenge, but a future one. We know how bad it can be if unchecked. How can we prevent future waves? Well, we really don't, but certainly global surveillance and strong science will be some of the key pillars of it. Thank you very much.
Thank you. Thank you very much. Really stunning numbers you presented there. Very good. So I think this concludes the speakers. So now we're going to open it to questions. Have we received any questions from the audience? Sean? Oh, and yes. Please open your cameras for the Q&A session, the speakers. Maybe while we get, yes, Sean, go ahead.
Thank you very much, Alejandro. You know, I do have a question myself. I mean, these presentations were fantastic and very interesting. I mean, Claudio presented on data, the exponential growth of data, risks and governance challenges. Wilbert provided an overview of Project Tycho. So, you know, I was wondering, maybe Wilbert could comment on steps that we could take to enhance international collaboration and open science around data to better prepare for global challenges of the future.
you, Sean.
Wilbert?
Yeah, thanks everybody else for their comments. See if I can speak. Yeah, I think, as I mentioned already, I think it's a complex landscape, and as I've mentioned as well. There are lots of data being generated all over the world by researchers, institutes, healthcare facilities, governments, etc. And so, as was mentioned by Claudio, the amount of data is increasing, the complexity of the data is increasing, but there's also, and so I think there is also no good vision for doing everything centralized. So, relying on one agency like WHO or one institute like NIH to gather up all the data, coordinate everything is unlikely to going to work. It needs, so I think the current thinking is mostly towards a federated way where repository, scientific, generalist repository.
Excuse me, if I could just butt in, Wilbert, could you lower your mic down? We're having a hard time hearing.
Sorry. Sorry. Yeah, that's terrific. So I was going to say that there is already a very complex landscape of data, and it's increasing rapidly. And having a centralized kind of place to deal with things is probably not the way to go. To distribute things as much as possible, so that researchers and agencies that do bring in data will be able to govern that and do that in their own way, but then make it in such a way that it becomes connected, or the ability to connect through use of standards, metadata, and when data are being shared or generated with the intent to say, how could others use this? How could this connect to other systems during the design of the data collection efforts moving forward? So I think there is no clear-cut one answer for this, but I think if we start to work at a global level with the different collaborative efforts, like WHO could set directions, funding agency could set directions when they're in conversation with each other, like forums like this, and also places like the Research Data Alliance or CoData or global organizations that think about data. If the directives and the recommendations that come out of there for standards, for interoperability, for metadata can be used when we design repositories across the world, then they could be brought together, searched together, and connect at some point. But I think we need to find a way where we will have some autonomy in different places to do what is best for their communities and their countries and their settings, but also at some point some conversations about how to connect them together so that the global community can much more leverage the different resources that are being built and not reinvent the wheels and duplicate things. But where to have that conversation and who to engage in these things, I mean, that's an ongoing discussion, I think, and various places are emerging of where to do that and how to do that.
Maybe others would like to comment. I know in Canada, we've had issues with even provinces sharing data. So I don't know if Prabhat wants to comment on that, maybe from his Ontario basis.
Well, I think one of the lessons that really has to be is you need federal systems. Canada was hit hard by SARS-1. David Naylor wrote at Jean Chrétien's request a brilliant report, and all of the recognitions pretty much were ignored, and one of which was to set up a national data portal. So it is extraordinary that and we need to adopt best practices. Right now, you can go on the Portuguese national mortality data set, and they'll tell you how many deaths occurred yesterday, and broadly by age groups and areas, and eventually the death certification. It's an online system, and in this day and age, not having those kind of real-time systems seems almost criminal, particularly for advanced economies. So I think these are the part of thinking about what we build would be the access to the vast amounts of data that governments collect in a much more timely 21st century and not a 17th century way, which is how we're doing things currently.
Now, I heard in your presentation, you mentioned that Peru had done a good job. Maybe our colleagues from Peru.
Oh, I think so. Yeah, I think Peru is to be commended for both bringing out early data that had problems, but then bringing out a more complete set of data which sorted out making the data available for researchers. So it's a very good example.
Perhaps Rafael would care to comment on that.
Well, yes. I'm particularly not involved in the processing of data about COVID-19, but we are always trying to talk with the Ministry of Health to try to induce to open more the data. For the genomic surveillance also, it's important. Many of the genomic surveillance of the variants of COVID-19 are centralized on the National Institute of Health here. And we are trying to push up from other regions. We have the capabilities here in Amazonas. Arequipa is other hotspot. Cusco also, and they have capabilities and they have human resources for this. And but we need a little more decision for the data sharing. That is important, no?
Okay, thank you very much. We also talked about biomanufacturing. Pablo talked about that in Argentina. I know in Canada, there's a big discussion and a big thrust for biomanufacturing because Canada is not producing vaccines; they're all imported at this time. I don't know if Pablo, you care to elaborate on how you see those efforts, and other people jump in. Pablo?
Yes, well, yes, biomanufacturing is a critical asset for distribution and access of vaccines and biopharmaceuticals. I think that it's very important to work on complementarity between different countries. I think that it's not possible to duplicate capabilities, but in some stages, in other stages like fill and finish, perhaps more distributed infrastructure is necessary. And I think that it's very important to work on a pool of clinical studies, because this is the main barrier to fields of the region to achieve the objective of production. There are a lot of research developments, but without this critical at the region, but without this clinical study at third level, is very, very difficult to develop regional vaccines. And it's a question of priority, I think, to go from science to solution at the level of vaccines.
Okay, well, thank you very much. I think according to the schedule, I think we're out of time. Thank you very much for your fantastic observations. And this has been recorded, and so the information will be there for people to look at. And also you will perhaps be contacted by members of the audience. Yes, you wanted to say something, Rafael?
Yes, Alejandro, sorry. I would like to respond to Prabhat about a question.
Yes. Well, go ahead.
It's a very interesting question about the when he asked, do you find the spike-based antibody assays are affected by malaria co-infection? Yes, we are suspect also that we have an episode here during the first wave here in Peru, in Loreto. Loreto was very high transmission of the virus. Well, during the first 5 months of the pandemic, the only test was the RDTs. So many people in in Iquitos that is endemic for malaria and other diseases or other infection diseases, they have high values of antibodies. So, but then in other waves that we had, well, the second wave, there's another kind of another cohort of people that reinfect. But we don't know if the first stage of the antibodies were product of any other previous infection or other coronavirus or other diseases. We suspect that. We need effectively to do more studies about that. Here in Amazonas is interesting because the endemic population is in the native communities, and that native communities are very close. Culturally, they refuse the intervention. So, but we are planning some strategies to make an intervention with the authorities of the community to try to make a screening. That's the reason because we are acquiring the Luminix 200 to make a massive screening. So it's interesting. Well, we hope that we have some results for next meeting.
Thank you. Well, thank you very much for that fascinating response. It's great to have this discussion here in real time. So I do think now we're out of time. Thank you all. So you saw the main issues which were discussed: data, biomanufacturing, and of course the population itself, the mortality rates, and the use of artificial intelligence, other techniques, all of science approach to this terrible pandemic and future endemic, as we saw. I want to mention to the audience that the last webinar in this series, which will be on education, will be tomorrow. And you'll see there the information about it. I hope that you will tune in, that you found this one interesting, and that you will enjoy tomorrow's webinar too. I want to thank the speakers for their fantastic interventions and the great discussion, and FAPES and GRC. Thank you all. Goodbye.
Thank you.
Thank you very much.
Thank you.